Bismillahirrahmanirrahim. Another paper, June 24 paper. Now we're going to do the A-level biology. So this is paper 1-1, the first MCQ paper. This paper is for 1 hour 15 minutes. Now this has changed since the syllabus changed in 2022. So now the MCQs are much more difficult than the previous ones. Now coming to question number 1, you can see the question using the same magnification. Chloroplast. Okay, the diagram shows a stage micrometer scale viewed with an eyepiece graticule using a magnification of 200. So now this one with the dark one, these ones which I'm just going to show you here, this is now the stage micrometer. We know the measurement of a stage micrometer between two lines, it's 0.1 millimeter. And this is the eyepiece graticule. So 40 eyepiece graticule is equal to 0.1 millimeter. Why? Because the stage micrometer actually gives us the measurements, but the eyepiece graticule is just divisions, just a line. It's just a ruler or just a line with divisions on it, but it has no units. So 40 eyepiece graticule units, we call it EPG, eyepiece graticule units, eyepiece graticule units, EPG, is equal to 0.1 millimeter. Now 40 is equal to 0.1, 1 is equal to how much? 4. Why 4? Because it says use the same magnification. Chloroplast is measured at 4 eyepiece graticule units. So 4 will be equal to how much? But this is in millimeter. This is in millimeter. Then you multiply it by 1000 and you get the answer in micrometer. So the answer was 1 into 10 to the power 1 micrometer. So the answer is A. Now coming to question number 2, and I've given you two diagrams which are of course not in the paper, but I picked them up from the net and I have put these for you all to make you remember that a typical prokaryotic cell, which is a bacteria, can either be 1 to 10 micrometer, and a typical eukaryote can be 10 to 100 micrometer. Now here you can see, I've given you this for a special reason, is an HIV or a virus is only 100 nanometer. E. coli can be, E. coli as a bacteria can be up to 2 micrometer. The red blood cell is 6 to 8 micrometer and a lymphocyte is 6 to 9 micrometer. Now here is another comparison. This is a total prokaryotic cell, typical prokaryotic cell. So it's 1 to 10 micrometer. 1 to 10 micrometer. 1 to 10 is the range. So they can be as small as 1 and as large as 10 micrometer. So that is why the answer in this case in the question number 2 is B, because it says which range of cell diameter is typical for prokaryotic cells. Now you have to know this, and it's something you've got to understand, that typical prokaryotes will be 1 to 10 and typically eukaryotes will be 10 to 100 typically. But you can see this red blood cell is 6 to 8 micrometers, so they're saying typically 10 to 100. So, but that's a very tiny cell, but as compared to the lymphocyte, which is of course 6 to 9 micrometer. So please remember this is in micrometer, not in nanometers. Now this one you see is in nanometers. So please be very careful in understanding whether it's nanometer or micrometer. You all overlook this many times and then make a mistake in this. And this is another table which I would like you to go through. And I would like you to understand that this is a very good comparison between, you see the prokaryotic cell here, you can see it is an average 0.5 to 5 micrometer, and this is up to 40 micrometer. And then of course all these other things, ribosomes, prokaryotes only have 70S and eukaryotes will have 70S and 80S. That's a defect in this table. So mitochondria absent, endoplasmic reticulum absent. Please pause the video here and have a look at this and write it all down. And please remember it. Now coming to question number 3, the diagram shows a typical animal cell. Where would nucleic acid be found? Now here comes the ambiguity. Nucleic acids are what? DNA and RNA. And RNA is mRNA, tRNA, and rRNA. Now where is ribosomal RNA found? In the ribosomes. tRNA will be found on the ribosomes in the cytoplasm. mRNA would be found in the nucleus and then it will migrate to the cytoplasm where it will get attached to the ribosome. So why would one have nucleic acid? One is the cytoplasm. It contains free ribosomes. It also is involved when the tRNA is coming here and when the mRNA is coming here. So it will always have nucleic acid in it. 2 is the ribosome, it has ribosomal RNA, rRNA. And 3 is the mitochondria, where it has circular DNA and it has ribosomes which have ribosomal RNA. So the answer for that is why, is A. 1, 2, and 3, all 3 will have nucleic acids in it. But you all forget it, and that is, I don't want you to be forgetting it. Which structure is found in a typical bacterial cell? Intron is not found, telomere, template strand, and capsid. So the template strand is the only thing which is present. Introns are only present in eukaryotes. Telomeres are linear DNA. This is circular DNA. Capsid is found in a virus. So it is typically confusing you that if you are confused between bacteria and virus, well, you can't do this question. Fine. Now coming to question number 5. What is the maximum number of hydrogen bonds that can form between a single water molecule and other water molecules? Now I want to share a diagram with you. Now as you can see here, this is a little confusing. It says what is the maximum number of hydrogen bonds that can form between a single water molecule and other water molecules. So the answer is D. There can be 4 hydrogen bonds with other water molecules. Question number 6. The diagram shows the structure of 4 amino acids in aqueous solution. Which two structures have an overall charge? Now look at the charge. Now I want everybody looking very carefully at the charge. This is a negative, this is a positive. This is a negative, this is a positive charge. Now here you see there's a negative here, there's a negative here, and there's a positive here. So this is 1. This is a negative here, a positive here, and a positive here. So this is 1. So lysine and aspartate, which is 6, the answer is C. Now coming to question number 1, it took me a little while to figure this out. And I'm sure some of you are very smart and you could have figured it out in the first go. It took me a little while to figure this out. I accept that, my intelligence level is not as smart as you all are. You're all Gen Z. Gen Z is a smart generation. The table shows the number of carbon atoms and the number of carbon-carbon double bonds in molecules of 3 triglycerides. So triglyceride will have a glycerol which has 3 carbon in it. 1, 2, 3. Plus it says, and the fatty acids they contain. So these are 3 different triglycerides, 1, 2, 3. The information shown in the format is number of carbon atoms and number of carbon-carbon double bonds. So the triglyceride is 54. So why would I say 54 minus 3? Because you see the 3 is from the glycerol. So you remove that, so this should be 51. So 15 plus 18 plus 18, and this is 1. Is 1 double bond, so the 1 double bond is here. So this 15, this 18 plus 18, how much is it going to be? 1, carry up 2, 3, 4, 5, so 51. So 51, 54 minus 3 is 51. So 51 plus 3 is 54. And there is 1 here. Then let's look at this one, 57 carbon. So 57 minus 3 will be 54. So 18 plus 18 plus 18. So that would be 54 and plus 3. So this 57 is 18 plus 18 plus 18. So this 54 plus 3 is 57. So we've got this right, and we've got 3 here. 3 were what? The number of carbon-carbon double bonds. So 3 double bonds here. So this is 1 double bond and this is the 2 double bonds. So the 3. Now let me give it another color so that everybody is very clear on what am I talking about. Let me just color these for you so that I know some of the students will find it difficult to comprehend it. So this is 3, that means 3 double bonds, 3 number of carbon-carbon double bonds. So this is a double bond 1 and this is 2, so these are 3. Now they've said which description matches fatty acid 2 of the 56,4. So we are now talking about, let me give it another color. We're talking about this one, which is a 56 and a 4. So the first thing you see is this is a 2 here. So there has to be a 2 here. So that means it's not saturated, it's unsaturated. And then 18 plus 17, and then you have 56. So 56. Now let's clear up all this and let me show it to you here again on another scale. So 56 minus 3 will be, so the total should be 53. Now we have a 17 here. So 17 and 18, 35. And then 53 minus 35 will be what? 8, 18. So this has to be a 18 2. Why am I removing the 3 is because the glycerol has 3 in it. Please remember I explained this earlier. So it has to be unsaturated with 18 carbon. It has to be D. Unsaturated. Why? Because it's got 4 double bonds. So it's unsaturated. And this is an 18. 18, please remember 18, 17, and another 18 here. I hope I've explained this and I hope everybody's understood it. So the answer is D, unsaturated with 18 carbon atoms. Question number 8. Collagen molecules are made up of 3 polypeptide chains interacting together. 3 polypeptide chains, 1, 2, and another 3. So 3 polypeptide chains, 1, 2, 3. The individual polypeptides consist of a regular pattern of amino acids. Glycine is every third amino acid. Almost every third amino acid is glycine. Which protein structures of collagen are described? I mean, in this story, in this question, when we've read the question, what 3 things have been? So it says a regular pattern of amino acids, and every third amino acid is glycine. So that would be the primary structure, which is the number and sequence of amino acids, right? So, and then we say it has 3 polypeptide, more than 2. So the answer is B, which is primary and quaternary structure. Primary and quaternary structure, because its quaternary says 2 or more polypeptide chains. Primary tells you the number and sequence of amino acid. But nowhere has it talked about the alpha helix and the beta pleated sheet. So secondary is wrong. We have not talked about. It says which protein substances are described. What is described means what has been written in the question here, described. Then coming to question number 9, the masses of the parts of hemoglobin are shown in the table. There are 1000 DA in 1 kDa. Masses in DA. So the masses of the parts are shown. So there's alpha globin is 15126 and beta globin is 15868 and the heme group is 617. What is the mass of a hemoglobin molecule in kDa? And this is DA. And DA is what? 1000 DA in 1 kDa. Now coming to question number 9. The masses of the parts of hemoglobin are shown in the table, alpha globin, beta heme. So we've done that. So then I'll show you the working of it, because there are 2 alpha globin and 2 beta globin chains. So multiply it by 2 and then multiply, because the heme groups will be 4. So you multiply it by 4, so you get this, and you divide it by 1000 to get the answer in kDa. So this would be D, 64.5. Then coming to question number 10. What is the correct order of location in the cell for the production of an extracellular enzyme? Extracellular enzymes are thrown out of the cell, and these will be made on the ribosomes, and these will be made on the fixed ribosomes. Of course, that's not in the question, but intracellular enzymes are made on the free ribosomes. So question number 10, what is the answer? The answer is A, nucleus ribosome. First, the nucleus, there's going to be transcription, then translation on the ribosome, and this ribosome will be on the rough endoplasmic reticulum because it's in the extracellular enzyme, and then it will go to the Golgi body, where it will be modified and then it will be thrown out of the cell. So, exocytosis. Then question number 11, the graph shows the trend from an enzyme catalyzed reaction, X and Y. Which labels are correct for the X-axis and the Y-axis? Y-axis, I would have sort of thought it will be rate of reaction, so I'm looking at that, and that is this one here. So the answer is C. It has to be pH and the rate of reaction, and nothing else fits in this because Y has to, something has to increase and then decrease, and X, something has to change. So if the pH changes, or we could have temperature and the rate of reaction. So that is why the y-axis, I would always look at the y-axis first and see what could it possibly be. So question number 11, the answer is C. Then coming to question number 12, which statements of the Michaelis-Menten constant Km are correct? The higher the Km, the higher the enzyme affinity for the substrate. Km is a measure of the degree of enzyme affinity for the substrate. Km is defined as the substrate concentration at which the enzyme functions at half of its maximum rate. Now the answer is D, but why is the answer D? You see, why is one wrong? It says higher the Km, higher. No, that's the opposite. It's inverse. It's not exactly inverse, but we say the higher the Km, the lower the enzyme affinity for the substrate. So this is the one which they've given wrong. This is a the first thing that we talk about is that the higher the Km, the lower the affinity. And that is how we do these experiments to find out if the amylase that we've taken from starch and the amylase that they've taken from rice, which has a higher Km. So higher the Km, lower the affinity. So we have to look at those amylases which have a lower Km. Lower Km, higher affinity. The diagram shows how nicotine is transported from the blood plasma into a cell using a type of co-transporter mechanism. So we've got the blood plasma, we've got the cytoplasm of the cell. So this is the cell membrane, which they've shown you here. And then they've shown you a co-transporter protein, and they say protons move down their concentration gradient out of the cell. So this is the one which is down, the protons are moving down, so they're moving into the blood plasma, down their concentration gradient. And then what is happening the other way around, nicotine is transported against the concentration gradient into the cell. So this is going into the cell, and then this proton is moving down their concentration gradient out of the cell. So protons would be higher here, so they would be 100 here and 10 here. So because down their concentration gradient. And this is again, so it would be the other way around. This would be 10 here and 100 here, and so it's against the concentration gradient. In the phloem tissue, there is a co-transporter mechanism that moves sucrose into the cytoplasm of a companion cell. Now they're comparing it with that. Which statement correctly describes a similarity between the co-transport of nicotine and the co-transport of sucrose? So you need to revise that. Hydrogen ions pumped out, hydrogen ions flow back, and they bring along with them sucrose. Now, a quick revision of this chapter. Co-transport, active transport of a solid. Now you see this is the proton pump. Now this is pumping hydrogen out. So this is a separate pump which pumps hydrogen out. And then the hydrogen ions diffuse back in and sucrose is co-transported. So there are two protein channel proteins. One is this one and one is this one for the co-transport of. So the hydrogen ion, when it diffuses back, it brings along with it sucrose. So one proton pump and the second co-transport protein. So co-transport, active transport of a solute indirectly drives transport of another solute. Plants use gradient of hydrogen ions generated by proton pumps to drive active transport of nutrients into the cell. Now let's go back to the question. When you read this, you realize why A is wrong. The co-transporter proteins generate a proton gradient by moving protons out of the cell by active transport. No, it's not the co-transporter protein which pumps the hydrogen out. It's another transporter protein. It's just a normal which pumps hydrogens out. So it's not the co-transporter protein. Now B is correct. The protons are transported through the co-transporter protein by facilitated diffusion. Now, why is C wrong? Let's look at that. The protons move through the co-transporter protein. Again, that's wrong. And then the co-transporter protein uses energy. No, it's facilitated diffusion to transport proteins with nicotine and sucrose. So they use energy from ATP. Actually, the hydrogen ions are pumped out, but then this does not require energy. The core uses energy to transport protons with nicotine and sucrose. So that is why C and D are wrong. Then coming to question number 14, the diagram shows a simple metabolic pathway W to X, X to Y, Y to Z. The letters W, X, Y, Z represent four different substances: starch, maltose, glucose. At each step in the diagram, the substrate undergoes a chemical reaction catalyzed by an enzyme. So everything, this is another enzyme, this is another enzyme, this is another enzyme. The reaction produces the next substance in the pathway. They're just like I said, starch, maltose, glucose. Of course, we'll have to come with a fourth one, so we probably have to come up with something else like proteins into tripeptides and then dipeptides and then into amino acids. Which statement correctly describes the enzymes taking place in the metabolic pathway? So each one is a different enzyme. They're all globular proteins, that's right. They all contain hydrogen in their structure, so that is why the answer is C. They all have the same tertiary structure. How can we say that? We don't know the number and sequence of amino acids. They're all different enzymes. One is an amylase, one is a maltase, or one is a protease, one is a peptidase. So then they can't have the same tertiary structure. So this was wrong. So it's only 1 and 3. Now, question number 15, a student observed the effect of two different concentrations of salt solution on blood cells. The student added each concentration of salt solution to one of the two microscope slides, and then a small drop of fresh blood was added. Each slide was viewed using the high power lens of a microscope, and the student's observations were recorded. Slide 1, no red blood cells were visible. What has happened? Water is moved in, the cell has swollen up, and then it has burst. So no red blood cells were visible. Now, why would water move into a red blood cell when it is less negative? So in slide 1, it will be less negative, minus 10. Inside is minus 100. So water moves in by osmosis. The cell swells up and bursts, and you can't see any cells. They're just pieces of the cell membrane, and you can't see them. Slide 2, what has happened? The red blood cells are visible to look slightly crinkled. So water has moved out of it. So it will be more negative outside and less negative inside. Water has moved out and the cell has shrunk, and as you can see something like this, which is the red blood cell has shrunk. So that is the answer is B. Now this is a very good diagram explaining this. Isotonic solution, nothing happens. Hypotonic, if cell is placed in a hypotonic, it will take in water across the membranes leading to hemolysis or lysis. Hypertonic, what will happen? If a cell is in a hypertonic solution, the solution has a lower water concentration at the cell cytosol, and water moves out of the cell leading to crenation, not cremation. Please remember that. That's when you burn the dead body. So this is called crenation. This is called lysis, hemolysis or lysis. You can remember that. And isotonic, of course, remains the same. Question number 16. Which statement correctly describes facilitated diffusion? Now, the answer is B. The process using channel proteins or carrier proteins and may or may not change shape. Uses energy is always wrong. That's not facilitated diffusion. It is a passive process, does not require energy. Use energy, wrong. And it's not in the opposite direction. Doesn't move ions in the opposite directions from a high concentration to a low concentration. Question number 17. Which row is correct for stem cells? They cannot repair cells. That is, I always tell you when I'm teaching you this, cells cannot be repaired. Tissues can be repaired. So it has to be a no. And 17, the answer is D. Can be involved in the formation of phagocytes. Yes, stem cells in the bone marrow will result in the formation of white blood cells or phagocytes or lymphocytes. So the answer is D. Then coming to question number 18, eukaryotic organisms grow and increase in size as a result of cell division. The diagram shows some stages of mitosis with one stage missing labeled X. So this is metaphase and then this is telophase. The answer is D because they are describing anaphase between metaphase and so, but they're not asking the stage, they're asking you. So microtubule shortens, the spindle fibers contract and the centromeres divide. So metaphase, the chromosomes have to separate. The sister chromatids must separate. So the centromeres divide. The centromeres are holding the two chromatids together. And now they're going to move to the opposite pole. So metaphase, then anaphase, they're going to move apart. A for a anaphase, A for apart. Metaphase is middle. Telophase is when the two nuclei form and the nuclear membrane would reform again. Question number 19. The graph shows various distance measurements from the start of metaphase of mitosis. The graphs are to scale when compared to one another. Now you can see X, the distance was, say, I always say put figures on it. Say the distance was 5, so there remains 5 and then it's decreasing. And it's going down to, say, 1. And here if you put figures 1, 2, 3, 4, 5, we see that at time this one, it was, the distance was 1 and then the distance is increasing 1, 2, 3, 4 and then it stays constant, say at 5 or 6. This is how you explain graphs to yourself. Then it says which row correctly identifies the distance measurements for each graph. So what was happening in X? It remained constant and then it decreased. Now, of course, now it's a little difficult if you have to really, if you've not understood the chapter on mitosis. Now, the centromeres, distance of the centromeres to the spindle. So this is the cell. This is the, you see, chromosome. This is the centromere. Centromeres to the spindle. So what you've got to understand is how this is going to change. Now, the answer to this is C, distance of centromeres from the poles to the spindle. So this distance of course remains the same and then it is going to decrease because you see the centromeres split and then this is going to move here, here, here and finally is going to reach here. Then this is going to move here. So the distance from the centromeres remains constant and then it starts to decrease. So this distance is going to, this was the distance originally and then this is going to become less and then less. And this other was the distance between sister chromatids. What does it mean, distance between the sister chromatids? You see, this is the, originally this was the distance. Let me give it another color. So that this is one centromere and this is the other centromere. And then slowly what is going to happen, distance between sisters. So this one is going to go apart and this one is going to go here down. So what is going to happen to the distance between the sister chromatids? So this distance initially was this. Now it is this. I know it's a very difficult question. It's a very difficult question. So distance between sister chromatids. So please try to understand it. It's technically difficult, yes.